Okay, so today we're going to be talking about our third class of biological macromolecules, which are called proteins. Um, Proteins are a very diverse group of biological macromolecules that are going to be important for us to understand a lot of the concepts that we're going to be learning from today on. Concepts such as evolution, genetics, cell biology, and even the human body systems. All of these concepts are going to be enhanced by understanding different types of proteins and their very diverse or different functions. So if we were to talk about proteins and their function, I can't really pinpoint a specific function for all proteins because they are so diverse. But if we were to discuss the basic function of proteins, they are important molecules in cells and in organisms that determine the structure and the function of cells. So they don't provide us with energy. They are not energy molecules. Instead, they help provide us with structure for our cells, and they also provide very, very important functions for our cells that if they were not there, we would not be living. So the elements of proteins, like lipids and carbohydrates, they are organic, so they have carbon and hydrogen. They also have oxygen, nitrogen, and sulfur. The one of the elements they do not have is phosphorus, um, but they have the other five. A little ahead there. So just a few examples of the diverse types of proteins and their functions. We have motor proteins. Motor proteins are these little molecules in cells that actually function in moving materials around the cell. So they move organelles, they move other molecules, but they actually function and they move and have like almost like little people that walk along the cell and carry things. Okay, they're like the UPS drivers. Okay. Another type of very, very important protein that we're going to be talking about are enzymes. Okay, this is really important. Put a star by this in your notes. Okay, enzymes are critical for understanding many of the chemical processes that go on in cells and in organisms. So it's very important to know that they are proteins and how they can be affected. The next one is antibodies, which are used by our immune system for defense from foreign substances such as bacteria and viruses. So when we talk about the immune system, we're going to be talking about antibodies, but they are also an example of proteins. Hormones such as testosterone or estrogen are cell signaling molecules that are used for the communication of cells, and they also lead to different developmental patterns in organisms, such as if you have higher testosterone, you will be developing into a male, whereas if you have a higher estrogen levels, that cause you to develop into a female. Cell surface receptors. Cell surface receptors are, again, important signaling molecules because they receive and transmit signals to the nucleus inside the cell. We're going to be talking more about those a little bit later. And lastly, we have structural proteins, which provide structural support for cells and for organs. So these are just a few. There are thousands and thousands of types of proteins with different functions. So again, you can't pinpoint any specific function for for proteins in general. However, you do know that they are extremely important for a lot of the processes that go on in our bodies. If we were to look at the monomers of proteins, okay, they are called amino acids. And they were called amino acids because they have two groups. Okay, so you have your amino group, which is this NH2 here. And you have your carboxylic acid group, which is where you get the acid part. So your monomers are amino acids. Okay, and they have, every amino acid has this NH2, and every amino acid has this COOH. Another important part about amino acid monomers is they have an R group here, which we'll talk about in a second, but an R group is different because there are 20 different amino acid monomers, and each of the 20 have a different R group attached to them. So here are those R groups. If you notice, you have a hydrogen can be an R group, CH3 can be an R group, or you can have as complicated R groups as you have down here. Okay, and you also have complicated R groups that look like rings, kind of like almost like a carbohydrate attached to it. It's not a carbohydrate, but it looks like one. So these are all R groups, and there are 20 different amino acids that can act as monomers for your protein chains. So when we talk about protein polymers, like a carbohydrate polymer, they are long chains of amino acids. Okay, polysaccharides are long chains of glucose molecules. All right, they kind of look similar to this when we draw them simply. However, not unlike sugars. Okay, these are your amino acids, and if you look, you have the different amino acids: glycine, isoleucine, valine, cytosine, glutamine. Okay, etc. And you can have thousands and sometimes millions of different combinations of these because they can be anywhere between 20 amino acids long and 10,000 amino acids long. If you look at the actual polymer itself, proteins end up folding on each other, and they create this globular shape that you see here. That is actually going to be very important for the functions that they perform. So a dipeptide is what you call a dimer, okay, of two amino acids that are chemically bonded together. And your polypeptides are going to be your amino acid chains that have any more than two amino acids in a chain. So this would be considered a polypeptide because it has more than one amino acid bonded together. So when we talk about polypeptide folding, okay, there are 20 different amino acids that can be arranged in millions and millions of different ways. Certain R groups on those 20 different amino acids are going to attract to other R groups. This attraction, coupled with or combined with the different numbers of amino acids in each protein, causes the protein to fold on top of itself, giving each protein a very specific shape. And one important concept, okay, so if I was going to highlight something about this slide that you guys really need to know, okay, it would be that hold on, let me go back. If I was to highlight something that you need to know about this slide, it is that certain R groups attract to each other, causing proteins to fold on themselves, and that shape that they get is what determines the function of the amino acid sequence or of the protein. So millions of different arrangements leads to millions of different protein functions. Very, very important. So if you have questions, this sometimes can be a little bit confusing. This is what questions you're going to want to ask me tomorrow in class. So here's a little video showing you how proteins end up folding on top of themselves. So if you notice, you have a sequence of different amino acids. Okay, so right now you have probably at least 100 amino acids that are bonded in this particular protein sequence. And right now they're in just one simple chain. Okay, however, this is not how the protein ends up looking after we're all said and done. So certain parts of the protein are going to have attractions to each other, and those attractions are going to cause this protein to fold on top of itself multiple times, giving it a specific shape. The specific shape that you see okay, that they're going to depict in this little video is going to be the determining factor of what this protein is actually going to do, whether it's going to be an enzyme and digest things, or whether it's going to be a receptor molecule and send signals to the nucleus. All those functions are going to be dependent on the shape that you see. And each protein has a specific shape, and that specific shape leads to a specific function. So when we're talking about polypeptide formation, we're going to be basically using our simple dehydration synthesis that we should all be familiar with now. Water is going to come out of the molecule. Okay, you're going to get rid of your water, and when you do that, you're going to end up forming what we call a peptide bond. So a peptide bond is what you see here between the carbon and the nitrogen, and that will form between every amino acid in the chain, creating multiple peptides. So the name polypeptide actually talks about how there are multiple peptide bonds between amino acids. So this is actually a tripeptide because you have three peptide bonds in this particular polymer. So if you want to see a video of how that actually would look, okay, here's a real simple video showing that I'm going to take the water, okay, so the OH group off of the carboxylic acid and the hydrogen off of the NH2 group. That leaves an open bonding site on the nitrogen and an open bonding site on the carbon, allowing them to form this peptide bond right here. And this is what you would call a dipeptide, obviously, because you have two amino acids. So that is the new peptide bond between the carbon and the nitrogen. So if you have any questions, make sure you ask them, because this is a very important lecture, because, again, we're going to be learning about many different types of proteins throughout the rest of the year. So make sure that you're understanding the monomers, the polymers, and the basic functions of different proteins that we talked about. Okay? I will see you tomorrow in class, and have a great night.